According to the secretions formed by the epithelial cells, glands can be classified into three types: merocrine or eccrine glands. Secretions are coming into the lumen. No loss of any cell membrane. Salivary glands. Apocrine glands. The apical surface of the cell is broken and there is loss of cell membrane containing the secretions. Mammary glands. Modified sweat gland. Holocrine glands. The whole of cell is broken down, the cell membrane is lost, and organelles are also broken down to form the secretions. Sebaceous gland, associated with hair follicle in the skin. Here we can see the skin epithelium with hair follicles, sebaceous glands, and sweat glands. In the sebaceous gland, the peripheral cells are having intact cell membranes. Towards the secretory portion, there is loss. There is loss of cell membranes and cell organelles, all becoming sebum, being poured alongside the hair follicle to keep it lubricated. Therefore, sebaceous gland is a holocrine gland. Sweat glands can also be seen here. They have simple cuboidal epithelium lining. This is merocrine gland. Classification of salivary glands. Serous gland, parotid salivary gland. They have serous cells, which are dark staining cuboidal cells with central spherical nucleus. They have thin watery secretions. Mucinous gland, sublingual salivary gland. They have mucous cells which are lightly staining columnar cells with oval basal nucleus. These cells have an empty loop or a foamy appearance. They have thick viscous type of secretions. Seromucinous gland, submandibular salivary gland. There is presence of both serous and mucous cells. There is presence of a special structure called serous demilunae. Serous demilunae, it is a cap of serous cells which is sitting on mucous cells. Characteristics of a serous secretory cell. These cells have a polarized cytoplasm, basal cytoplasm. It is more basophilic, bluish staining. This is due to presence of rough endoplasmic reticulum towards the base, which are protein synthesizing organelles. Apical cytoplasm. It is present towards the secretory or luminal side. It is more eosinophilic, pinkish staining. This is because of presence of variously staining secretory vesicles containing zymogen granules. The Golgi apparatus is usually located midway along the cells, typically at supranuclear position. Ribosomes and endoplasmic reticulum makes proteins and pack them into secretory vesicles. These secretory vesicles joins the membrane bound stacks of Golgi bodies. These Golgi bodies repackage them to form secretory granules or lysosomes and send them towards the luminal side. These lysosomes fuse with apical cell membrane and secretions are sent out. Histology of skin. Skin has two main layers: epidermis upper layer, dermis lower layer. Epidermis can have 4 or 5 layers depending on location it is present in. The above skin specimen is taken from palm, thick skin. Therefore, we can see 5 layers of epidermis: stratum corneum, stratum lucidum, stratum granulosum, stratum basale, stratum granuosum. It's a lot of keratin fibers and dead cells. It is because of these keratin fibers that this layer has an eosinophilic appearance, pinkish color. Stratum lucidum. It's a thin pinkish layer. It's only seen in areas of thick skin like palms and soles. This layer has dead cells. Stratum granulosum. It's a keratohyalin granules for formation of keratin filaments. Stratum spinosum. It is also known as prickle cell layer. It's the thickest of all layers. It is called so because cells in this layer have spiny or pointed appearance. The spiny appearance is because of abundance of desmosomes in this layer. The desmosomes in this layer are comprised in diseases like pemphigus vulgaris, which causes skin blisters. Stratum basale. It is also known as stratum germinatum because this layer has stem cells that keep adding more layers of skin as it sloughs off from the surface. We will discuss